Welcome once again to my channel. In this video, I'm going to talk about photosynthesis, the process of photosynthesis. So basically, when we talk about photosynthesis, we are talking about the process by which plants manufacture their food, which is glucose, using carbon dioxide from the atmosphere, water from the soil, energy from sunlight in the presence of chlorophyll. So without carbon dioxide, without water, and without chlorophyll, you cannot have photosynthesis. So these are the essential components needed for photosynthesis to occur. And chlorophyll can be found in what we call the chloroplast, right? It's a chlorophyll that gives the leaf the green color. So let's look at how the chloroplast looks like. So this is a diagram of the chloroplast. If you cut across it, a cross section through the chloroplast, right? So as you can see, it has an outer membrane, which is called external membrane out here. There's the external membrane. They have an internal membrane here, and an intermembrane space, a space between the two membranes. And within the structure here, inside the membranes, is what we call the stroma. Okay, and within the stroma, you have so many things embedded in it. You have the chloroplast DNA, as you can see here, and then you have the thylakoids. These are the thylakoids. Okay, thylakoid. So they look like stacks of coins, and each coin structure is called the granum. The granum. Okay, the granum has a lumen within it. Okay, and the lumen, that's where we find the chlorophyll. So the chlorophyll can be found in the granum, the lumen of the granum. Okay, and then you have ribosomes and other things here, like the plastoglobule. So these are just a basic structure of the chloroplast. Right. So photosynthesis occurs in two stages of photosynthesis. We have what's called a light-dependent reaction. And then two, you have the light-independent reactions. So light dependent and then light independent. So one needs light and one aspect does not need light. So let's begin by looking at the light dependent reactions. So the light dependent reactions use photosynthetic pigments organized into what we call the photosystems to convert light energy into chemical energy, specifically ATP and what we call NADPH, nicotinamide adenine dinucleotide phosphate hydrogen. These reactions occur within specialized membrane disc within the chloroplast called the thylakoids, as we've just seen in the diagram. So the thylakoids contain you have the granule, right, within the thylakoid, making up the thylakoids, and within that you have the chlorophyll. So there are three steps involved here. One, you have excitation of photosystems by light energy. And then two, you have production of ATP via an electron transport chain. And three, you have reduction of the NADP plus and the photolysis of water, right? Photolysis of water. So that's breaking down of water using light. So three stages: excitation of photosystems by light energy. They produce ATP via the electron transport chain. And then you have reduction of the NADP plus. It means and then photolysis of water. Reduction means that you're adding hydrogen to the substance. When you add hydrogen to the substance, we refer to that as reduction. So the overview of the light-dependent reactions. This is what it's all about. So the light-dependent reactions occur within the intermembrane space of the thylakoids or granule, as we've said. And chlorophyll in photosystems 1 and 2 absorb light, which triggers the release of high energy electrons. We call this photoactivation. Now, the excited electrons for the photosystems 2 are transferred between the carrier molecules in an electron transport chain, right? So you have excitation of electrons. That's what is occurring here. Electrons get excited, right? So because of the high energy from the sunlight, okay, in what we call the photosystem 2. And then those electrons are then transferred from one carrier molecule to the other in the electron transport chain. So the electron transport chain is made up of carrier molecules that carry or transfer electrons. Now, the electron transport chain translocates hydrogen ions from the stroma. You know what the stroma is? You just saw that in the diagram of the chloroplast to within the thylakoid. And this creates a proton gradient, right? So that means as the hydrogen ions are moved from the stroma to the thylakoid, there is a proton gradient that is created, right? Now the protons are returned to the stroma via the ATP synthase. That's an enzyme, right? Which uses the passage to synthesize ATP, right? This is what we call chemiosmosis. So the protons that are returned to the stroma via the ATP synthase, right, uses their passage to synthesize ATP. So ATP is formed during this process as the protons are moved from one carrier molecule to the other, right? Now, the excited electrons from photosystem 1 are used to reduce the NADP, forming NADP plus, forming NADPH, right? So that means the NADPH is reduced to the NADPH. So this is coming from the photosystem 1. Okay, we just said there are two photosystems, 1 and 2. Okay. And this is the part that comes from the photosystem 1. You know, the electrons there are used to reduce the NADPH, DP plus, sorry. Now the electrons from the photosystem 1 are replaced by the energized electrons from photosystem 2. Now the electrons lost from photosystem 2 are replaced following the photolysis of water, right? Now the products of the light-dependent reactions, the ATP and NADP, are used in the light-independent reactions. So this is where we're going to see the connection between the light-dependent and the light-independent reaction. So the light-independent reaction is supposed to create ATP and NADPH that can be used in the light-independent reaction. So let's look at details of the light-dependent reactions one more time. Okay, so this is going to be more details of that. So step 1, we said that it's a citation of photosystems by light energy, right? That's what we just said. So the photosystems are groups of photosynthetic pigments, including chlorophyll, embedded within the thylakoid membrane. Now, photosystems are classified according to their maximal absorption wavelengths, right? So photosystem 1, which you call the PS1, shortened to PS1, they can absorb light around 70 700 nanometers, while photosystem 2, PS2, can absorb light waves around 680 nanometers, right? So the classification based on the wavelength of light and the light wavelength that they can absorb. Now, when a photosystem absorbs light energy, there's still light energy. The localized electrons within the pigments become energized, right? Energized, I mean, they become excited. Okay. These excited electrons are transferred to carrier molecules within the thylakoid membrane. So that's a step one, what occurs there, as I've just described, these are details. And step two, as we said, is the production of ATP via an electron transport chain. Right? So the excited electrons from photosystem 2 are transferred to an electron transport chain within the thylakoid membrane. As the electrons are passed through the chain, they lose energy, which is used to translocate hydrogen ions into the thylakoid. Right? Now, this buildup of protons within the thylakoid creates an electrochemical gradient, or what we call the proton motive force. Right? We mentioned that that's the chemiosmosis that we're talking about here. Okay. And then step two, you have part of a continuation of step two, you have production of ATP via an electron transport chain. So the hydrogen ions return to the stroma along the proton gradient via the transmembrane enzyme, ATP synthase, right? Now the ATP synthase uses the passage of hydrogen ions to catalyze the synthesis of ATP from ADP and organic phosphates. This process is what we call photophosphorylation. As light provided initial energy source for ATP production. Okay, the energy came from light, so called photophosphorylation. The newly de-energized electrons from photosystem 2 are taken up by photosystem 1. Okay. And then the step 3, the details of that, the reduction of NADP plus and the photolysis of water. So excited electrons from the photosystem 1 may be transferred to a carrier molecule and used to reduce NADP plus. This forms NADPH, which is needed in conjunction with the ATP for the light independent reaction. Right? So these are the details of what I just described prior to this. Okay. Now, step 3 continued. So the electrons lost from the photosystem 1 are replaced by the energized electrons for photosystem 2. And the electrons lost from photosystem 2 are replaced by electrons released from water by the photolysis, okay, or photolysis. Okay. Water is split by light energy into hydrogen ions used in chemolysis, and oxygen is released as a byproduct, right? So the byproduct that we see in photosynthesis comes from the breakdown of water. Okay. All right. So that was a summary. I did like detailed information on the light-dependent reaction. What are C4 and CAM, the CAM, photosynthesis? Okay, so let's talk about that, and then we'll continue with the other part, the light-independent part. Now, C4, carbon fixation, it's a form of photosynthesis in which plants concentrate carbon dioxide spatially with a ribulose biphosphate or bisphosphate carboxylase, shortened form to Rubisco. Reaction center in a bundle sheet cell that is inundated with carbon dioxide. So you have a lot of carbon dioxide in that space. Then you have the CAM, which is a Crassulacean acid metabolism. Crassulacean acid metabolism. It's a carbon fixation pathway that is involved in some plants as that evolved in some plants as an adaptation to arid conditions in which the stomata in the leaves remain shut during the day to reduce evaporation, but open at night to collect carbon dioxide. Okay. So photosynthesis in desert plants. In harsh, dry heat, every drop of water must be conserved for survival because stomata must open to allow for uptake of carbon dioxide. Water escapes from the leaf during active synthesis. Now, photosynthesis in desert plants has evolved adaptations that conserve water. A more efficient use of carbon dioxide allows plants to adapt to living in less water. Right. So some plants, such as the cacti, can prepare materials for photosynthesis during the night. They use a temporary carbon fixation and storage process during the night because opening the stomata at this time conserves water due to cooler temperatures. So in addition, cacti have evolved the ability to carry out low levels of photosynthesis without opening stomata at all, a mechanism for surviving extremely dry periods. So in addition, cacti have evolved the ability to carry out low levels of photosynthesis without opening the stomata at all, a mechanism for surviving extremely dry periods. I believe I've said this already. Then we have the xerophytes such as cacti and most succulents. They also use the phosphoenolpyruvate, the PEP, carboxylase to capture carbon dioxide in a process called Crassulacean acid metabolism. Can plants fix the oxygen at night? Sorry, carbon dioxide at night when their stomata are open. So basically, it's like a summary of all that I've said under the CAM photosynthesis. They have their photosynthesis at night to conserve water, all right? Now, the CAM plants store the carbon dioxide mostly in the form of what we call the malic acid via carboxylation of phosphoenolpyruvate to oxaloacetate, which is then reduced to malate. Okay, so that's a process how the CAM plants they store their carbon dioxide. Okay, then decarboxylation of the malate during the day releases carbon dioxide inside the leaves, thus allowing carbon fixation to 3-phosphoglycerate by the rubisco. Okay. So that's a very efficient process that the plants that we find in deserts use to help to conserve water. Now, C4 plants, with C4 carbon fixation. Now, the C4 plants, they concentrate carbon dioxide spatially with the rubisco reaction center in the bundle sheet that is inundated with carbon dioxide. Now, C4 plants can produce more sugar than C3 plants in conditions of high light and temperature. Now, many important plants are C4 plants, including like maize, sorghum, sugarcane, and millet. Okay, these are examples of the C4 plants. So they produce more sugar than the C3 plants in conditions of high light and temperature. Okay, high light and temperature. Now, plants that do not use the PEP carboxylase in carbon fixation are called C3 plants because their primary carboxylation reaction catalyzed by rubisco, produces the 3-carbon 3-phosphoglyceric acid directly in the carbon-benzene cycle, right? So they produce 3-phosphoglyceric acids in the carbon-benzene cycle. Over 90% of plants use C3, carbon fixation, compared to 3% that use a C4 carbon fixation. So a lot of plants use C3 process, C3 carbon fixation. So let's look at the carbon cycle. Now, the carbon cycle consists of 3 basic stages. You have the fixation, reduction, and regeneration. Fixation, reduction, and regeneration. Now, in plants, carbon dioxide enters the interior of the leaf via pores called the stomata and diffuse into the stroma of the chloroplast. Now, the stroma is the site of the carbon cycle reactions where sugar is synthesized. These reactions are also called the light-independent reactions because they are not directly driven by light. Okay, so now we can talk about the light-independent reaction because they're not driven by light. Okay. So let's look at process, the 3 stages. Fixation. Light-independent reactions are initiated during the fixation. Carbon dioxide is fixed to an inorganic, is fixed from an inorganic to an organic molecule, right? They had a reduction stage, where ATP and the NADP are used to reduce 3-phosphoglyceric acid into glyceraldehyde 3-phosphate, G3P, all right? Then ATP and NADPH are converted to ADP and NADP plus respectively. In other words, the NADP is becomes oxidized, right? That's what it's oxidized, it loses the hydrogen. NADPH loses the hydrogen to become NADP plus. Okay, that's what we're trying to say here. Then you have the regeneration. The regeneration, the RBP is regenerated, which enables the system to prepare for more carbon dioxide to be fixed. So in the stroma, in addition to carbon dioxide, 2 other components are present to initiate the light-independent reactions. There's the enzyme called ribulose bisphosphate. That's the rubisco. And 3 molecules of ribulose bisphosphate, the RUBP. And RUBP has 5 carbon atoms flanked by 2 phosphates. And rubisco catalyzes a reaction between carbon dioxide and the RUBP. For each carbon dioxide that reacts with 1 RUBP, 2 molecules of 3-phosphoglyceride is formed. And the 3-PGA has 3 carbons and 1 phosphate. Each turn of the cycle involves only 1 RUBP and 1 carbon dioxide and forms 2 molecules of 3-PGA. Now, 3 atoms of carbon dioxide plus 15 atoms of the 3-RUBP will give you 18 atoms in 3 atoms of 3-PGA. And this process is called the carbon fixation because carbon dioxide is fixed from an inorganic form into the organic form. So this is a diagram of what's happening in the cycle, okay, the carbon cycle in the diagrammatic form as we just described, okay. Stage 1, the fixation, right? 3 molecules carbon dioxide with a rubisco converts it to 3-PG. That's the first stage, carbon fixation. Then you have the 6 ATPs because you have 6 molecules here, you need 6 ATP to phosphorylate this. It gets phosphorylated. So the ATP becomes ADP, and then you have the stage 2, the reduction, where you have NADPH being reduced, right? So that's the process. Yeah. The summary of what is occurring in what I've just described. Okay. Right, so the ATP and the NADP are used to convert the 6 molecules of the 3-PGA into 6 molecules of a chemical called the glyceride 3-phosphate. Now, the 6 molecules of both ATP and NADP are used. For ATP, energy is released with the loss of the terminal phosphate atom converted to ADP, right? So this is describing the cycle that we just seen. For NADPH, both energy and a hydrogen atom are lost, converting it to NADP+. Right? Both of these molecules return to the nearby light-dependent reactions to be reused and re-energized. Okay. So it's a cycle. You use and reuse and reproduce. Only one of the G3 principles leaves the Calvin cycle and sent to the cytoplasm to contribute to the formation of one other compound needed by the plant. Because the G3P exported from the chloroplast has 3 carbon atoms, it takes 3/10 of the Calvin cycle to fix enough net carbon dioxide to export 1 G3P. But each 10 makes the 2 G3P that's 3/10 makes 6 G3PS. One is exported while the remaining 5 G3P molecules remain in the cycle and are used to regenerate RUBP, which enables the system to prepare for more carbon dioxide to be fixed. 3 more molecules of ATP are used in these regeneration reactions. Okay. So this is a summary of what happens in photosynthesis. I hope you enjoyed watching this. It's a little bit complex, but if you take your time to read, you would really appreciate it. Stay tuned for more videos.